Hi, my name is Lula Wang, and I'm a 10th grader from Greenwich High School. My research project this year was on the lipid-based co-delivery of doxorubicin and siRNA PDL1 as a multifunction chemoimmunotherapy selective to pancreatic cancer via its MUC1 overexpression. I became interested in this topic because pancreatic cancer is the second leading cause of death worldwide, with a 5-year survival rate of less than 10%. Pancreatic ductal adenocarcinoma, or PDAC, constitutes 90% of pancreatic cancers, which was therefore the focus for my project. Currently, the most widely used chemotherapy drug is doxorubicin, or DOX, which functions by blocking topoisomerase II, an enzyme that cancer cells rely on to divide and grow. However, what's unique about PDAC is that its dense stroma and aggressive tumor microenvironment, or TME, alongside the overexpression of MUC1 glycoproteins and PDL1, suppresses the immune system by preventing T cell function. This causes traditional anti-tumor drugs to have high toxicity and poor selectivity, causing off-target side effects such as nerve damage, heart failure, and even secondary cancer such as leukemia. My approach that I will introduce to you takes advantage of 3 things. Firstly, it uses the overexpression of MUC1 to give specificity and targeting. Secondly, it prevents the mRNA code for the production of PDL1 that removes immune response by incorrectly adhering to T cells. And finally, it uses the cancer cell's aggressiveness and lipid metabolism to obtain energy. This suppression of the immune system is due to programmed ligand 1, or PDL1, which is a protein expressed on PDAC cells. Here's an animation that I made, which illustrates the role of PDL1 and my project's approach that uses PDL1 targeting siRNA to resolve immune silencing. During proper functioning, T cells actively detect and destroy cancerous cells. However, in PDAC, the PD-1 receptor on T cells incorrectly binds to PDL1, preventing function. What I discovered from previous literature by Barati et al. Was that siRNA PDL1 can target and inhibit the production of PDL1, therefore blocking T cells and restoring immune response. Thus, moving forward, I incorporated this approach into my therapeutic. Therefore, I designed and engineered a novel multifunctioning therapeutic using a targeted chemoimmunotherapy nanoparticle approach through a series of steps. First, I constructed MPEG-B PHEP nanoparticle cores. These serve as effective centers by providing flow core capability, enhanced drug release, overcoming drug resistance, and allowing for the addition of layers while maintaining stability. I used ATR-FTIR for each additional component to provide evidence for coding, where the x-axis is the wave number and the y-axis is percent transmission. Next, I infused DOX with the MPEG-B PHEP nanoparticle cores. Here's PHEP, then I added DOX, and as you can see in the last spectrum, that there is clearly DOX in the DOX-coded nanoparticles. Moving forward, throughout each step for the synthesis of my therapeutic, I invite you to compare each of these 3 spectra, which provide evidence for continuous accession. To further provide DOX stability in the tumor microenvironment, I then added SiO2. Following the SiO2 separation layer, I added chitosan as the next external layer to provide opportunity to attach siRNA PDL1 to the nanoparticle. Knowing that immune silencing and chemotherapy resistance could be resolved by siRNA PDL1, I was determined to attach them to the chitosan layer so that it could do 2 things. Firstly, target and degrade the mRNA that codes for PDL1, therefore closing the channel of immune suppression. And secondly, act as a secondary way of passage by holding the nanoparticle near the cancer cell membrane so that it enters the cells for endocytosis. Then, I coated the lipid layer composed of DOPE onto the docked siRNA nanoparticles. DOPE is a lipid that is commonly found in the TME as part of cancer cell membranes. Therefore, by using a biomimetic approach, the DOPE lipid layer makes the therapeutic TME-friendly and stealthy so that it survives in the TME. Finally, to provide specificity to pancreatic cells, I coated MUC1 antibodies alongside the DOPE lipid layer to create the final layer of the completed therapeutic, which I named DOX siRNA DOPE. MUC1 is a glycoprotein overexpressed only by PDAC cells and not on healthy cells. Therefore, it would serve as a perfect target for the therapeutic, which would prevent off-target consequences commonly associated with traditional chemotherapy. Additionally, this model requires that these MUC1 are bound to the dope layer, and I showed evidence of it through FTIR, where the primary amine group of the anti-MUC1 attaches to the dope carbonyl group under slightly acidic conditions, therefore forming an amine bond. Here's a schematic that displays the coding process and a diagram that shows what my therapeutic involves now. The completed nanoparticle has MUC1 on the outside, the dope lipid layer, siRNA beneath it, protective coatings, DOCs, and finally the MPEG-B PHEP nanoparticle core. I used SEMs to display the coding progression of the nanoparticles, starting from the MPEG-B PHEP nanoparticle cores to the finished DOCS siRNA dope therapeutic. Similarly, I use dynamic light scattering to further highlight the increase in particle size throughout the layering process. I am now going to show you the functionality of every aspect that I built. First, the anti-MUC1 on the nanoparticle surface binds to MUC1 overexpressed on PDAC cells. Then, the dope lipid layer degrades, taking advantage of the TME's aggressive desire to break apart lipids, also known as lipid metabolism. This releases the DOCS siRNA nanoparticles, which are then free-flowing and target PD-L1 through passage, silencing its gene expression and therefore restoring immune response. The therapeutic is then taken in through endocytosis, releasing DOCS, which triggers apoptosis for the destruction of the tumor. Since I needed to provide evidence for the functionality of the therapeutic without having access to pancreatic cancer cells because I was working in my high school, I had to design a creative approach. To start, I had to provide evidence that my therapeutic targeted MUC1. What I did was I bought an MUC1 ELISA kit, which normally sandwiches reagents for the detection of MUC1. My thought was that I could use the anti-MUC1 and MUC1 standards that come with the well and see if my nanoparticle could adhere to them. I then used this as an opportunity to trap the MUC1 standard to show that my therapeutic binds to it. So in the first one, you notice that I have a spectrum of plastic from the bottom of the well that I flipped over, anti-MUC1, which I had because I happened to use it during the construction of my nanoparticles, and then on the bottom, you could see the anti-MUC1 as purchased in the well. This is important because I knew I could use the material in the well and I could see the material in the well and therefore could use this IR technique moving forward. Following, I loaded MUC1, I washed it, and as you can see, it is bound to the anti-MUC1 on the bottom of the well plate. Finally, I loaded my therapeutic on it, and after washing it again, I could see its presence, which is important because it highlights the successful specificity and binding to MUC1. Following confirmation of successful targeting to MUC1 on PDAC cells, I now had to verify the pH-specific and timely dissolution of the dope layer. Therefore, to first simulate the pH-specific lipid degradation, I added the nanoparticles to 30 microliters of a solution with a pH of 7.4. I then decreased the pH by 0.1 increments until it reached 6.8 to mimic the change from normal extracellular fluid to the pH level of the TME. At those 0.1 pH increments, I took out 10 microliters of the solution, centrifuged it, and measured the FTIR spectrum after the solution dried. Using the peak area for total dissolution, I confirmed 100% dissolution of the dope layer to be at a pH of 6.8 in the TME. Now that I knew that the dope layer could degrade in the TME, I did a similar experiment where this time I timed the dissolution at a pH of 6.8. I then used the IR in the same way as before, and the dope layer took 5 minutes to dissolve, confirming complete dissolution prior to endocytosis. Now that I've confirmed successful targeting and dissolution of the outer dope layer, I then needed to determine release efficiency of DOCS. Therefore, at 250 nanograms per milliliter, I used serial dilutions, and here's a calibration plot that I created. I then compared it to the peak area for DOX release at 2.73 minutes, and the results highlighted that at 5 minutes and a pH of 6.8, 0.76 nanograms of DOX was released. This represents a 95% release efficiency, which is an almost 100% improvement from current therapies, with a release efficiency from 30 to 50%. This is significant because it means that my engineered therapeutic has a much higher delivery rate, therefore decreasing dosage amount, which minimizes toxicity to the body and cost of treatment. Finally, I wanted to mimic normal versus artificial cancer cells with overexpressed MUC1, so I conducted a final experiment by constructing giant unilamellar vesicles, or GUVs, with separate lipid membrane compositions. After adding the completed DOX siRNA dope therapeutic, the results were concluded using fluorescence microscopy images of the simulated cancer cells in comparison to the simulated healthy cells. Throughout 300 seconds, the artificial healthy cell membranes remained intact. However, the membranes of the artificial cancer cells with MUC1 overexpressed showed signs of membrane disruption at 150 seconds, and at 300 seconds, they were intermixed entirely. This highlights the DOX siRNA dope's successful function and specificity to PDAC cells following lipid fusion. To summarize, I have designed and developed a therapeutic that overcomes all of the shortcomings in current delivery and takes advantage of every characteristic and bad habit that PDAC cells have that make it so difficult to treat. In the future, I plan to apply this engineered therapeutic to many other cancers as well, with the addition of specific targeting mechanisms to their overexpressed proteins. I'd like to thank JSHS for giving me the opportunity to present and share my research. Thank you for listening, and I'd be happy to answer any questions.